Great. So thanks, everyone. Glad to see everyone back for the spring semester or the fall semester. This is how quick it is. And, thank you to the Qualcomm community for all the work putting everything together. And it's good to see lots of people here. So it's my pleasure today to introduce Dr. Oliver Beckstein. And so he's a colleague in our department. He earned his bachelor's degree in 1999 at the Friedrich Alexander University in Erlangen-Nuremberg, and then his PhD in 2005 at the University of Oxford and at the Laboratory of Molecular Biophysics and Biochemistry, working on sort of early MD analyses. And what grew out of that in some ways is a research career that's sort of centered around also open source software and making community tools available, and then using those to find advancements in sort of fundamental biophysics. And so after his PhD, he spent some time both as a research fellow and a postdoctoral fellow at Oxford and also at Johns Hopkins. And he joined the faculty here in 2012. And I think probably we can leave it there and let him take it away and tell us about wiggling molecules.
Okay. Thank you very much, Doug, and thank you for leaving some time for talking. Does this microphone work? So, okay, cool. Okay, so it's great to see you all at the beginning of the semester. So let me start with a quote that you've now already seen a couple of times in this colloquium. We're fully aware that this is probably the fourth time that you're seeing this in 2 years. But I think there's a pretty good reason. When Richard Feynman said, everything that living things do can be understood in terms of the jiggling and wiggling of atoms. And you as physicists probably say, that's kind of trivial. I mean, we do this all the time. We think about the fundamental interactions between particles and think about how these sort of basic interactions sort of combine into a bigger picture. So I think for physicists, that's actually quite a natural way to look at things. But then thinking that sort of complicated sort of realm of living things has to obey the exactly same rules that we do in our physics labs, I think that is something that, again, physicists probably appreciate, but the rest of the scientific world, might actually take some time sort of getting to. The other reason why I think this quote, shows up quite a bit is because, in a way, it provides a program sort of to do biophysics. The idea is really that if I start at the bottom, then I can build up an understanding of the fundamental processes of life, from the little things that we can understand, that we can easily probe. And this is ultimately what I want to talk about today, sort of our lab's effort in how we can get from the wiggling, jiggling of atoms to understanding biology, at least to some degree. But because there's also a colloquium, I don't want to just dive in directly. I want to take a little bit of time of setting up the stage and talk to you about, what is this realm of biology that we're trying to understand. So for a moment, just imagine that we want to, that we need an engine like this one, car engine. So, and of course, what do? We, get some blueprints and then we need parts. But wouldn't it be wonderful if we could just sort of sprinkle some fairy dust over these plans and then sort of lean back and see these plans sort of fold themselves into this, this engine that we need? And then you might get stuff that looks like this. It might be a complicated process, but at the end of the day, you might get sort of something fairly complicated that has sort of self-assembled from these sheets of papers that had the instructions on them. So I hope I see some sort of faces that sort of squint, and say, really? Well, I mean, this is all totally made up. I just used the stable diffusion generator and asked it to, give me whatever I needed for this talk. So this is total rubbish here. So of course that doesn't work. You can't just have a blueprint that folds itself into a machine. Or can we? Because if you look at a different scale, now we're going to the molecular scale, and we have proteins here. So a protein is a string of different chemicals, amino acids, that are organized in a specific sequence. And this sequence is essentially the blueprint for this protein. And as you can see here in this animation, this protein folds, it assembles into a very well-defined structure. So unlike what I showed you before, that is actually based on real science. I mean, there's still computation involved and animation and so on, but that is actually encapsulating how we understand how proteins form at the moment. So what you can see here in the back is another sort of big protein machinery, the ribosome, that essentially connects these different amino acid residues. And this whole process takes about 15 seconds or so, and it happens gazillion of times in all of you every minute. So what you can also see here is sort of the little sort of atoms shown, white are this white, probably hydrogens, and there's all kinds of other stuff in there, carbon, of course. Sorry, Siri, I really don't want to talk to you. So why does this? Why do proteins fold? So in this animation, you can see the polypeptide chain again, and a little bit more clearly, you can see that there are different residues or amino acids that have different chemical characteristics. And it's exactly these sort of 20 different characteristics that are important because they encode different interactions. So the interactions that ultimately fold this protein are encoded in the sequence. And Kendall and others came up with this idea that what the sequence really encodes is a funneled energy landscape, so that over time, the protein, when it sort of randomly moves around, explores this landscape and ends up in the stable folded conformation. And what you should see here is that what looked initially like spaghetti now has sort of a very well-defined shape. So of course, here we've enhanced sort of the patterns inside the proteins by drawing this so-called alpha helix. So this is a common motif in folded proteins, this sort of corkscrew kind of parts that act like rods. And then the other things, these are beta strands that make sort of flat sheets. And these are the two fundamental secondary structure building blocks of proteins. And all of this is literally encoded just in how you string these amino acids together. So we do have, machines that self-assemble. But so the take-home message here is that the overall structure of proteins is sort of a key thing, and it is already in there. So why does everything wiggle about? So that is because all of this happens at the angstrom nanometer scale. It all happens in solvent, so you have water molecules moving around, as shown in this simulation here. So that means that when you have other particles, they are sort of pushed around by these water molecules and everything else. So we have these thermal motions. And most of what really happens on timescales that are longer than the so-called collision time. That's basically the time that a water molecule needs to reorient. And that's sort of picoseconds, give or take. So, but what we typically have to look at these length scales is these random motions. And not the typical sort of ballistic motions where you just, that you've been doing in your physics labs. So as an example, if you want to look at very simple things, so for instance, how far a particle sort of can move away from the origin, which is Brownian motion, then you observe this particle many times and you get different trajectories where you plot the position of the particle. Over time, you get this sort of, mess here, but there's actually sort of very simple laws hidden in there. So if you do some sort of averaging, then you can immediately see that there's a linear relationship between the squared distance from the origin and the time. So even though I'm not going to talk very much about diffusion here, I just want to make sure that everyone understands that even though it all looks very messy and everything jiggles around, there are fundamental laws, and that is really what statistical mechanics is about. So but within all of this moving around, we have to sort of, we have to contend with one thing, and that is that the typical energy scales that we are looking at are on the order of thermal fluctuations, which are measured in kBT. And, this is about 25 millielectron volts, so that's not a lot if you're, doing a lot of other physics. Another way to talk about this is it's about 4.1 piconewton per times nanometer. And the typical mechanical work that biological proteins do is on the order of 10 piconewtons times nanometer. So the mechanical work, basically, whatever the proteins actually need to do in your body is not much stronger than just this sort of all this wiggling and jiggling about. That means all the time there's this competition between random motions all over the place and actually, motion that accomplishes something. So one of the key questions then in molecular biology is, what do proteins have to do in order to survive this kind of sort of environment? So as an example here, I want to show you a movie that starts out by looking at a cell. So the first sort of two parts of this movie are actual experimental data. So this is a real fluorescence image that shows the nucleus of a cell. And so you can barely see sort of the cell membrane. And the worm-like things are mitochondria. So these are organelles inside the cell where essentially the energy of your body is being produced. We are going to zoom in further. So we're now at a sort of 100 nanometer scale, and we'll go even closer to the sort of tens or so nanometer. And this is now an animation. This is not experimental data anymore. But what you should be looking at is this ATP synthesis enzyme and ATP synthase. And you can see that there are differently folded parts, subunits of this protein, and all of these undergo sort of very specific motions. It's these motions that catalyze a fundamental reaction in biology, and that is forming ATP, which you might know as sort of the energy unit of the cell. What's key here is that the motions undergo through a certain cycle. You can see very well-defined states. If you look long enough, you see that each of these blue parts of the protein just does the same motions over and over again. And these cyclic motions are ultimately powered by a rotary motor, because if just everything would be randomly moving around, then you couldn't have any of these directed motions. But instead, you have this motor in the membrane that is powered by some source of free energy. In this case, the source of free energy is the fact that there are many more solvated protons, which I'm just going to call protons or H+ from now on, but it's fairly complicated what they really are in solution. So there are many more protons on the inside than on the outside. So that means that there's an electrochemical gradient. And in this gradient, the cell stores energy, and it releases the energy by essentially having the protons move through channels in this rotor and then driving these rotary motion. So the key takeaways here is that proteins need structure in order to function. And the way that they do their job is going through cyclical processes that use the source of free energy. So I put an asterisk on there because Sarah typically calls me out on this. Whenever you're talking about biology, there's probably an exception. So that's sort of one thing that I learned by now. So when I say that all proteins are folded and have sort of a well-defined structure, then this is a blatant lie. So that is simply not true, because there are many very important proteins that are called intrinsically disordered protein, and that don't follow this paradigm. And this is a very interesting area of research. I'm just going to completely ignore it here. And if you want to know more, talk to Sarah and a few other people who are doing this. So I'm going to talk about proteins that can change their shape, can change their conformations, these cyclical processes. And I now want to sort of jump into sort of the specific area where my lab has been doing some work, and that is just asking the question, how does stuff get into and out of a cell? So this is the textbook, literally the textbook picture of a cell. And I highlighted the plasma membrane in blue. That's what divides the outside from the inside. It's essentially a fundamental part of any cell. So, but this sort of boundary is actually pretty tight. And so normally not much can go through. This boundary is only 4 nanometers thick. So we zoomed in dramatically from sort of the micrometer scale now to the nanometer scale. And this boundary is made from lipids, another important biomolecule, and it essentially does not let anything through. That's, again, a bit of a lie, but mostly anything that the cell really cares about is probably not going to go through in the amount that it wants it. So what does a cell do? Well, they are so-called integral membrane proteins that act as gatekeepers in the membrane. And there are different types of these proteins. We are interested in so-called active transporter proteins that you can think of as pumps. They can move molecules against a gradient. So they are molecular machines that use some source of free energy. These transporter proteins are important in many different areas. So Here, for instance, in neurotransmissions, we have a synapse. And once the nerve impulse arrives, neurotransmitter is released, the little sort of green blobs here. They diffuse across the synaptic cleft, and then sort of a new action potential sort of is started. But of course, the neurotransmitter here can't just accumulate, because then you would stop transmission. So there has to be a process that take the neurotransmitter up back in, and these are these uptake transporters. So the word reuptake might be something that we've heard before, because the current class of most used antidepressant drugs simply act on these transporter proteins and block them, leading to an increase in neurotransmitter concentration in the synaptic cleft, in particular dopamine. Even though this process sort of acts within minutes, it then over the time scale of weeks or so leads to complete changes in mental states. I think this is a really good example of how things at the micro scale can really have an effect over different time scales and different length scales. And so it's clearly important to understand how stuff like this works if you're in the area of sort of drug discovery, for instance. So other areas where this is important is uptake of nutrients in the gut. So food comes in here and it has to go through the epithelial, the cell in your gut into your bloodstream. And there's one transporter, this SGLT, which transport glucose, sugar, and it's powered by sodium. We'll talk about this a little bit more about what powers these transporters. But what's interesting is that this transporter also more or less accidentally moves a lot of water molecules, about 50, 60 water molecules whenever it transports one sugar molecule. It's not clear if nature needs this. However, probably one of the most life-saving sort of drugs that has ever been invented is basically electrolytes. So when people die from dehydration due to diarrhea, you give them electrolytes. And the reason is simply because the sugar and the salt, in the electrolyte make these transporters work over time. And so a lot of water gets accidentally just put up back in the bloodstream. So again, sort of an example where understanding these sort of micro details might actually help sort of to make life better. And then there are other sort of important ones, and I'm just going to skip through those. So let me hone in sort of a little bit more on sort of the area that we've been working on. So this is a reasonably realistic picture of a transporter, except that I've shown these secondary structure helices as these sort of sausages, but they are essentially drawn from an X-ray crystal structure. So basically, experimentally, we have a good idea where all the atoms inside these transporters sit. What you can sort of see here is that we have two different conformations of this transporter. The wheat or brownish area here does not really change relative to the cell membrane, but this blue region here is in this down conformation and this up conformation. So my colleagues, it took quite a while until they got these two crystal structures, and I don't know. I thought I put somewhere else I have the citations, but the first structure that was 2013, now 10 years ago, the other structure was 2016. So I've been sort of playing around with these proteins now for 10 years. And so today I'm going to tell you sort of what we really know about them so far. But the key point here is we have these two conformations. How does this fit into what I've been talking sort of before? Well, I told you in order to do work, we have to have cycles. So let's draw sort of a physicist sort of view of what this sort of biological cycle looks like and put in what we know. We know these two structures. We call this an outward facing open conformation because a sodium ion can actually sort of exchange with the outside and the protein, but it can't get through. You need to actually switch the conformation, the shape of the protein, to go to this inside conformation. And then the sodium ion could sort of exchange with the inside. So we need other states in this cycle. So we need a state where this outward facing conformation doesn't have the substrate, the sodium ion bound. Then we need to bind a driving ion. So the driving ion is essentially the proton. Remember how in the video I told you that there's a gradient of protons. These transporters use exactly the same gradient. So you have to imagine that you have a surplus of protons on the outside. So there's a thermodynamic driving force for these ions to flow to the inside of the cell. And what this transporter really does, it provides them a way to go into the cell, also going from by switching this conformation, but without really opening up a leak. So you can imagine that the cell spends a lot of energy just establishing the proton gradient. I mean, there's not a spontaneously forming proton gradient. In fact, this is why you digest food, and the electron transfer chain does all this work there. So establishing these gradients is a huge amount of work. So if you just essentially drill a hole through the membrane and let the proton leak through, then you're just wasting all of this. So that's why this sort of airlock or switching, or it's called alternating access mechanism, is crucial, so that you don't have a leak of your driving ion. And then you go around the cycle, and once you've done this, when you keep going around the cycle, then you're doing work, because you're moving sodium ions from the inside to the outside, which happens to be the function of these kind of proteins. So I sort of drew these pictures, but let's try to understand a little bit better why this actually should be working. And it also allows me to introduce a little bit sort of terminology. So let's start in a very, very special thermodynamic state, namely equilibrium. Equilibrium is when macroscopically nothing seems to be changing. I mean, there's sort of a common joke. So what do you call a cell in equilibrium? A dead cell. So because life happens out of equilibrium. But let's start there. So what defines equilibrium is this condition called detailed balance. So we can understand it in the following way. Imagine that the system is in state 1, outward facing, with this driving ion bound. You have a certain probability P1 to be in this state. And you have a rate K12. That's essentially telling you how many transitions you can have per unit of time, sort of going from state 1 to state 2 if you're in state 1. So then the flux, or probability flux, is simply probability to be in this state times the rate to get out of the state. Detailed balance now says that for any kind of states that you might think up, it has to be true that forward and backward fluxes have to be the same. And this is actually a very, very sort of stringent condition. But most of what we teach in statistical mechanics is mostly, in this very special state. So what happens if we're moving out of equilibrium? Imagine that you're now sort of putting a lot more protons on the outside. Now you're setting up a gradient. In a way, you're increasing the probability of this state. So the flux from the state 1 to state 2 is now actually higher than it would have been if you had been in equilibrium. So that also increases the probability of state 2. And I'm sort of glossing over a little bit of the details here, but effectively what happens now, you can think of this in the same way, that this state probability is too high compared to equilibrium. So it keeps sort of pushing the cycle in this direction. And because the cycle is connected, which is really the key here, you start driving the cycle around in a circle. As I said, driving circles is the way to fight, all the randomness that's around you. What you should realize is that you can also drive the cycle in other ways. You don't have to play around with this ion. If you increase the concentration of the ion down here, then you can also drive it. Or if you increase the concentration of the sort of blue thing here out there, cycle around in the other direction. So it just works all the way. Just to mention that there's a special non-equilibrium state called steady state, and that is when the state probabilities don't change, but the fluxes are not the same. And this is actually what we typically sort of work with, but that's sort of a bit of a detail here. So, but once you drive the cycle, then you're doing work. And in this case, the work is moving sodium ions. And you can even move sodium ions against the gradient. As long as the driving ion gradient is stronger than the gradient that you're working against, you do work. And what's also interesting is that in one of these transport cycles, you have a fixed stoichiometry. So that means that if you're sort of a biochemist and you think about enzymes, and this should remind you of enzymes. What these transporters actually are they effectively enzyme for a transport process. So how does this give us a way to sort of do more quantitative things, beyond just sort of drawing these sort of little squiggles? I introduced the rates, and a simple model that we can use is called a master equation or a rate model. Essentially, all that we're doing is we're adding up all the in and out fluxes in this model and relate this to the changes in probabilities. Given if we have these individual rates, these are rate constants, they are specific for the system, but they don't really change. And the normalization of the probabilities, then we can solve the system of equations in order to get the state probabilities and the fluxes. And ultimately, the fluxes are the thing that will tell us about how many cycle completions you have per second. That is essentially our readout for function in some form. So what I'm saying is that sort of our approach for following the Feynman sort of wiggling and jiggling of atoms to function is that we want to start with structures which we get from experiment, and then we want to use computational approaches in order to get at these rates and maybe free energy difference, and then using physics-based simulations, and then use these master equations in order to get at the overall dependence of the processes on different parameters. Should also mention that you can also go backwards. So instead of bottom up, you can go top down. And that is when you then use measurements, especially from experiments, but we can also use measurements and then infer parameters for these models. So that's also an interesting area, but I'm not going to talk more about this today. There is a technical problem, and I only sort of put this one slide in there, and that is because these different parts of the cycle don't all happen at once. We actually have to divide and conquer. We have to, analyze one part of the cycle and another part of the cycle, and that can be this in experiments. It can be the same. Yeah, so that means that you obtain parameters from disconnected experiments or disconnected simulations. But there's sort of a hitch, and that is that these different parameters actually have to still obey certain equalities. So in particular, detailed balance that I mentioned before is effectively central to all of this. And I'm not going to unwrap it. I'm just going to say that when you're in equilibrium and you go around in a cycle, then the sum of free energy differences has to be zero. That might be immediately clear to you because free energy is a state function. But there's also a non-equilibrium equivalent for this, which is also not hard to understand, but is initially perhaps a little bit surprising, that relates the rates directly. So basically, products of rates around any cycle in the forward direction divided by rates backwards have to be 1. And that has to be true for any complicated cycles that you draw. So if you have really simple models, then that's easy to do. But if you have models with many subcycles, then this gets really tricky. And so Ian came up with a way that we can solve this problem, basically where we can ensure that we have these thermodynamically correct rates. So I already mentioned that we're interested in sodium proton transporters. So there are 2 types of these transporters in prokaryotes, basically bacteria. These are typically of the so-called CPA2 family. So what they do is they use protons as driving ions. And what they do is they move sodium out. It helps the bacteria to live in high salt environments like our stomach, for instance. Some plants also do the same thing. And then they can live in brackish water and so on because they can desalt their cells. So it can actually be interesting to know more about this in order to grow salt-resistant varieties. In humans, the so-called CPA1 families transporters use sodium ions as the driving ions and then move out protons. And they are essentially housekeeping proteins that are important to keep the pH of your cells constant. And there are a bunch of diseases that are related to this. And that's what I put in my NIH proposals. Oops. Oh, that's the same. Basically, that's the same slide. So I said that for NAPE, we know that 2 protons are moving in and 1 sodium is out. And the structure, we have 2 structures. We have this outward-facing conformation and the inward-facing conformation. And in this electrostatic plot here, you can So in this electrostatic representation here, red means negative potential and blue means positive potential. So the ions that we, I just say this right, red means negative, yeah. So the ions that we have are cations, they are positive. So negative means that they're actually attracted to getting into the protein here. So and in particular, you can see this sort of bowl-shaped depression here, cut through the protein, where a residue called D157, aspartate 157 sits. And this residue sort of switches its accessibility from the outside to the inside. So this is the most likely sort of key binding residue. And there's also mutational data that supports this idea. We know from, as I said, we worked on this sort of for a couple of years. So we have a pretty good idea of what actually happens in the binding site. And because I think sort of less than 1% of you, are deep into the field of sodium proton antiporters, all I want you to do is believe me that we have a good idea of what sort of the key parts of this binding site are so that we can study them in detail. Just want to point out there are these two aspartate residues and this one lysine residue that will play major roles. And there has been some ongoing debate in the field of who exactly is doing what in here. And as I said, there are probably 10 people in the world, who fight with me on this in papers, and we write papers back and forth, and it's all in good fun. So, but there is, the nitty-gritty details matter at the end if you want to do this, what I'm talking about here. So the transport cycle that we have set up, you have now seen this a couple of times. The only variation is really that I drawn two protons here. And what we need to do is we need to somehow understand how protons bind and how sodium binds and unbinds. These are key steps in the cycle. And we need to know how the conformational change happens. So what we really know is just we have these two sort of conformations, and that's about it. And the idea is then, if we know all these bits and pieces, we can put a full rate model together and learn about the transport. So coming back to Dr. Feynman. So I think what he was really hinting at is that if we know the wiggling or jiggling of atoms, as in the position and momenta of all these particles, basically the phase space coordinates, then we can use the tools of statistical mechanics to basically know everything about the system. So it's a little bit more complicated, when you say want to do non-equilibrium things, but that's sort of the very basic idea here. So how on earth are we going to get, this sort of detail? How do we know where all the atoms is? Luckily, there is a tool called molecular dynamics simulations, and classical in this kind. So what we're doing here. And the idea is that we can parameterize the energy of the whole system as a function of all the coordinates, and n can be 100,000 or 1 million or something like this. And we have a good idea of what these different terms are. Then the force on the ith atom is simply minus the gradient with respect to this atom. And then we use Newton's second law in order to set up a system of coupled ordinary differential equations. This is pretty annoying and nasty because this is a function of, as I said, 100,000 parameters or something like this. So we use big computers to solve this, but then we get trajectories that we can analyze. So the plan here is we do MD simulations for the binding. We basically directly observe sodium binding and unbinding. Protons are difficult because you can't directly simulate them with these classical simulations. So we do something quite special. It's called constant pH MD replica exchange simulations. And I'm going to completely gloss over it, and I'm happy to answer questions. And for the conformational change, normally you have to do really hard work there because these are so-called rare events that you normally don't see in these MD simulations, because the timescales that we can typically sample with MD simulations are far too short compared to these conformational changes, unless you're lucky, as in this case, and you're working with a thermophile protein that is actually quite fast when you elevate the temperature. In this case, we could actually do that. So Ian and Jack sort of did this, but I just want to sort of show you what these results look like. And so first, if you can look at these time traces here. So this is a time scale of 14 microseconds. So that's reasonably long for the kind of simulations that one can do. So I would say the current normal standard for these MD simulations is about a microsecond, or give or take. but sort of beyond 10 is long. And with these sort of traces here, you can see that they're from the distances that sodium ions sort of come into the binding site, leave again. These are actually different ones. So we see plenty of these kind of events. So once you have these events, you can do simple Boltzmann statistics and calculate the binding free energy as the fraction of bound to unbound states. Additionally, and that's not shown here, from these time traces, you can also get kinetics. So you can basically calculate the off rate, which is something that we'll need for later. So it's not that easy to compare these things to experiment. In the case of NAPE, at least our collaborators managed to measure the binding affinity in the inward-facing conformation. And sort of the experimental number is about 1.6 millimolar or minus 6.4 kT. And we are getting about minus 4.2 kT or 15.4 millimolar. So that's actually pretty good when you're doing this kind of classical MD and you're not really putting anything else in except the structure. So from this, we have an idea of how the sodium behaves. As I said, the protons are kind of difficult, but there's an interesting observation here that is going to be important later, is that when a sodium ion binds, it comes near these two residues. So aspartate 156 has a negatively charged carboxylate group in red, and lysine 305, K305 has a positively charged amine, and they form a salt bridge, basically a Coulomb interaction. Once the sodium ion comes in, it actually pulls the aspartate 156 away. And that is shown in this plot. When the sodium D156 distance is small, then the salt bridge distance is large. So that means the salt bridge breaks. That has the effect that the lysine here sort of moves away, and the positively charge on the lysine is not stabilized anymore through the Coulomb interaction. So the free energy of binding this proton drops. The biochemists would say that the pKa of this lysine sort of drops, but the effect is effectively that the proton comes off. So the sodium can compete away one of the protons. That is going to be key in the overall mechanism by which the sodium proton antiporters exchange sodiums and protons. It's something that comes directly out of these calculations, sort of shown in these shifts here. I don't want to go through the details here, but I need to tell you that the free energies that we get from the MD and from the CPH MD and the pKs and go into one of these models that I briefly mentioned before. And effectively, what we then do is, once we have consistent energies and pKs, we solve the partition. We have the partition function on this graph, and from the partition function, then we can get everything else that we want. In particular, we can get the effective binding free energy as a function of external parameters like pH and sodium concentration. And when we plot this, so the simulation data sort of at the top here, the binding free energy of a sodium as a function of pH sort of drops down. When it drops below 0, then binding is favorable. If it's above, then it's unfavorable. And at least qualitatively, these results compare with well with the experiments. So this is isothermal calorimetry data, which effectively shows that at low pH there's no binding. That's sort of the red region. And then at a pH 8.5 or higher, we actually have pretty strong binding. Basically, what this means is when there's little protons, high pH, then the sodium can actually get into the binding site and stay there. Otherwise, at low pHs, many protons, the protons outcompete the sodium. So qualitatively, that sort of gives the right answer. So that is encouraging. So, but now what does this conformation transition look like? And as I said, there we just got a little bit lucky because all we really had are these sort of two structures and a reasonable hypothesis of where the protons go. As I said, there's been debate in the literature and between people in this field, which of these, where this proton binds. And as I already alluded to, we think that this lysine is one of the proton binding residues and not two aspartates. And we prepared our system sort of according to our hypothesis. And gratifyingly, what we observed in very long MD simulations, and this is just a simulation which has water, lipids, and everything, but for clarity, I removed it. I would like you to focus on the sort of cyan left-hand side. We have this sort of light blue domain, and you can see that it sort of rotated a little bit, sits there for a while, and then it sort of seems to be sliding down. So I'm not forcing it to go anywhere. This kind of movement is encoded in the structure. Yeah, so it just does this once I parameterize everything, as I've been parameterized the 100 other membrane proteins that I've been sort of working with. This kind of movement is spontaneous. It's essentially an equilibrium movement. That's okay. In equilibrium, you can have as many forward as you can have backwards reaction. So there's nothing that says that in equilibrium you can't move. You can just have to move forward as much as you move backward. So this is actually exciting because this is not something that people normally see. And in fact, there are essentially, I think, 2 other examples in the literature at all where you have simulations like this that actually show you a full conformational transition. We then sort of looked at how to characterize these structures, and want to show you that when we start out, water can come in from the outside. Once we are sort of at the end of the simulation, the water accessibility switch to the inside, and there's this intermediate state, which we call an occluded state. That is also exciting because many of these transporters do have these so-called occluded state. It's just we've never seen one for these sodium proton antiporters. So it might be too transient to actually see in crystallography, but I'm talking to my colleagues if there's a way that we can sort of trap this, which would be really nice. We've did other sort of tests to show that where this simulation goes is actually very, very similar to the crystal structure, the inward-facing crystal structure that we have, which was just never part of the simulation. So there's really good evidence that what I've been showing you here is a relatively reasonable representation of how this protein goes from an outward-facing to an inward-facing conformation. Of course, this is n equals 1. That's a little bit bad. So we did a lot more simulations sort of starting from there and eventually harvested a couple more. So these are not good statistics by any means, but it's actually much better than what you normally get. And the advantage of having at least some statistics, you can then use simple Bayesian estimates that uses all the data, including the many that didn't really do anything, to arrive at a reasonable estimate for the rates. And what you can see here is that the conformational change rate is consistently only about 100 per millisecond. So this predicts that on average per millisecond, I see 100 of these transitions. So 1 every 10 microseconds or so. What you can also see is that the barrier crossing time, basically how long it takes for the protein to switch, is relatively short. All right, so we then put everything together, and I think I have to speed up a little bit. So this is simply saying that we start putting all the rates into a model. Some of the rates we simply don't know. Protons are really difficult. So we have to estimate the on rate and then sort of use the pK to estimate the off rate. So there are some issues here, which I sort of come back at the very end. But we then build sort of a big model. And again, don't worry about reading all of this. The take-home message here is that once you have a model, you can ask the model what it wants to do. And what it wants to do means this sort of big red line. That's the maximum flux through the model. So we start, for instance, if we start here, that's the outward-facing conformation with 2 proton bounds. And the movie that I showed you is sort of this. But then there's sort of a decision point. What do I do with the proton? It says, well, the first proton that has to go, given all the data that we have, is the one on D157. And then the model again would have a choice. You could lose the second proton, but what instead happens is the overwhelming sort of pathway is the one where now the sodium comes in and then outcompetes the proton. So what's really nice here is that instead of me having to make up a story of what all the bits and pieces mean, I actually get a sequence of most likely events. And at the end of the day, that is very much sort of what we consider a mechanism, right? If this happens, then this happens. But this other thing can also happen. This model sort of gives you everything there. And you can then basically have an annotated landscape here. You can also then look at the actual how fast this sort of cycle runs and plot this as a function for different parameters. So in this case, we set the inward pH to the outward pH and then drove it by a sodium gradient, the model, and you get this kind of normalized activity, which shows what is very characteristic for these sodium proton antipodals. On the left-hand side are experimental data, and they show the same kind of behavior. You have sort of peak activity around 8.5-ish or so, and no activity at sort of low and high pH. So the explanation is actually quite simple. At low pH, you have so many protons that they crowd out the binding site and no sodium can come in. So that is already what we saw with the binding. At high pH, there are just not enough protons to drive the cycle. So that's not really a thermodynamic effect, that's a kinetic effect. So it's simply that you don't have enough to go through that part of the pathway, and that simply slows down everything. So we can then also look at proton-driven transport. So unfortunately, I don't have data for NAPA, so this is a related protein that shows the overall kind of shape that we get. Again, we get all of this without fitting any parameters, right? So we just started with the crystal structure, and we know the diffusion coefficient of the proton. So that's what goes in there, and everything else is just in the model. And finally, we can calculate the overall sort of transport rate as a function of membrane potential and sodium concentration. And what you can see is that quite robustly, this protein or this model can move sodium against the gradient, which is positive turnover, even if there's a relatively high out concentration. So even if the concentration of sodium on the outside is 1 molar, it can still shuffle sodium against this gradient, and the inside concentration is 10 millimolar. The only thing that's really a little bit sad is if you look closely at the absolute value, that's only 2 turnovers per second. That's actually way, way less than we roughly estimated from experiment. So that's 2 orders of magnitude at least off. Luckily, I'm going on sabbatical this year, and my homework is actually figuring that out. And because we think that the key problem here is how we treat protons, because that's the thing that we know least about. So one thing I'm going to do is do ab initio simulations of protons and simply look into this a little bit more. But the good thing about a model like this is that you now have places to look at. You can say, I think there's a problem in the model here. Is this a process that we don't understand well enough? And then you can go in and do more research. So in summary, I basically showed you how you can go from structure to function using physics-based simulations as the generator of the dynamics, and then use these very simple kinetic cycle models in order to arrive at some macroscopic predictions that you can at least start comparing to experiment. So with this, I just want to say thank you for coming out, and to all members of my group that were involved in this project. Particularly Ian has done a lot of the work here, but everybody else has also been contributing important points. And my experimental collaborators at Stockholm, David Drew and Jana Shen's group, with whom we worked on these CPHMD simulations. And thank you to various funders. Thank you.